Thanks, Graham. And also, I want to take the opportunity to thank Adrian and Susan and others who are driving this project along. It's pretty amazing to have so many New Zealand researchers focused on this particular research theme. Anyway, to cut to the chase, for a while now, I've been interested in how rat populations recover after 1080. And this afternoon's talk focuses on some of the work that I'm currently doing, looking at the influence of treatment size, shape, and geography on patterns of recovery, rat recovery, particularly ship rats, using the data from the DOC rat tracking tunnel database. Why are we interested in patterns of rodent recovery? Well, ultimately our goal is to protect and restore populations of native birds, bats, lizards and insects. And this depends primarily on two things. Firstly, controlling rats and other key predators to near zero, as Josh has just been discussing, over very large areas. And second, after control, slowing the recovery of rats and other predator populations as much as possible to create a large time window where native animals are not exposed to significant predation or competition. As we get better at controlling rats over large areas, immigration will increasingly drive rodent regrowth after control, and as a consequence will become an important area of focus for us if we seeking to maximise the persistence of control. So what do we know already? Well, some time ago we did some work in the Tararua looking at rat tracking rates across a treatment boundary. So the treatment in this instance is this grey shaded area at the centre of the Tararua's Project Kaka, which is surrounded by these light grey areas of no treatment. So we were monitoring tracking tunnels over these treatment, along transects over these treatment boundaries. And we found the pattern of rats tracking before and after control sort of reflected a pattern whereby tracking tunnels closest to the treatment boundary increased first after control, and then later tracking rates further into the treatment area increased. And when we came to model these patterns, we found that an interaction between time since treatment and straight line distance best explained this pattern. However, a straight line distance isn't really it doesn't seem reasonable when thinking about the way rats might travel over landscapes where there are mountains and lakes and so forth. In our particular study, we were focused, in the Tararua, we were focused on two valley floors with no topographical impediments of any note. But normally, we might be looking at places with high mountains or large lakes, and so it seems reasonably unreasonable to think that rats are going to travel across those landscapes in a straight line. Probably do what we do and follow the path of least resistance. So what might this path of least resistance look like? Well, some work by Jenny Christie and Susan and others has shown that rat tracking generally declines with increased elevation, and rat tracks are relatively infrequently recorded above 1,000 metres. So we can sort of assume from these bits of work that areas of high elevation act as an impediment to rat movement. And similarly, work by James Russell and others has shown that rats can swim between islands, but that ship rats are somewhat reluctant swimmers. And islands separated by more than 100 metres can have genetically distinct populations on them, suggesting that that sea barrier between those islands acts as an impediment to movement. So the hypothesis on which this talk is based is that rats don't travel in straight lines, but wiggle themselves around features of the landscape that they find difficult to traverse. So how do we test the influence of these geographic features Well, thankfully, there's a pretty well advanced area of study called least cost modelling or cost distance modelling. It was originally developed for transportation planning, but it's increasingly used to model the relationship between populations and the geographic context in which they occur. So the general principle is that you can create or represent geological and ecological features on the landscape as cost surfaces, like these matrices illustrated here in this slide. And then you can calculate the least cost path through those surfaces and tot up the distance traveled along that path and the accumulated cost. The other thing that you can do with these cost surfaces is you can add them together, so you can have a cost surface for lakes and rivers, a cost surface for high mountains and so forth, and test the relative influence of each of those different surfaces or topographical features on rodent growth post 1080, which is exactly what we set out to do. So with the help of Josh, I pulled out of the rat tracking data set 178 operational boundaries and linked those with tracking tunnel data. I then whittled these operations down to only include those that had 3 years post-control data, more than 5 data points, and only selected ops that had high kill rates, because I'm really interested in what happens in operations where kill has been very effective. Anyway, this left a grand total of 95 operations and linked tracking tunnel data. We then also sucked into a New Zealand DEM, a lakes and river surface, and also a barrier surface for sea and areas over 1,000 metres. And then in a very large loop in R, I extracted each of the tracking tunnel, the treatment boundaries, which are marked in this figure in this light grey, and linked those with the tracking tunnels, which are here marked in light blue lines, and created a 150 metre buffer around the tracking, around the treatment area, and then created a series of potential immigration origins, which are marked as blue dots. I paired these back so there were no immigration points above 1,000 metres in the middle of lakes or in the sea. And I also excluded immigration points that were in exclusion zones, so areas that Tenehi wasn't applied to because there was a hut or a track or so forth. If those zones were greater than 300 metres across, any sort of point of origin was removed. And so I had this suite of origin points and treatment boundaries, and then using the rasters I imported into R, created 10 cost surfaces, which were all based off the cost surface in the top left, which penalise sea and areas over 1,000 metres 25-fold. So for every grid square crossed in those areas, you'd suffer a 25-fold penalty. And then I created from that basis a series of cost surfaces with increasing cost for crossing lakes and rivers from 2, 5, 10, 25-fold on the top of this figure. And then in the bottom added an elevation penalty, so a 1-fold cost for every elevation, 100-metre elevation increment gained. And then through these cost surfaces, using the G-distance package in created a series of shortest paths from those points of origin on the treatment boundary to each of the tracking tunnels in the treatment. So you can see the 2 different cost surfaces here. The one on the left is one with a low penalty for rivers and lakes, and the one on the right is one with a high penalty, and you can see how it affects those shortest paths. I then whittled those short paths down to those only in the 5th percentile and calculated the mean distance of those paths from the treatment boundary to tracking tunnel. and also the mean accumulated costs and the cost of traversing all of the different pixels on the way from the treatment edge to the tracking tunnel, and appended both those values to each tracking tunnel in the data set. Finally, for each treatment polygon, I created a Euclidean distance surface, which sort of represents a straight line distance between the treatment edge and all grid cells within the operational area. I guess you can think of this as representing a scenario where rats could immigrate from all points on the treatment boundary. It's somewhat unlikely, but nevertheless, it was a useful contrast for the least cost metrics, and was what we used in the Tararua study. And I wanted to look at how this advanced approach related to that. And so then each of these Euclidean distances were added to the tracking tunnel data. And I also calculated a mean Euclidean distance for each treatment area as a sort of a proxy for edge to area ratio. So finally, after brutalizing my home computer for about 17 hours, I ended up with this data set to model. And I did this in a two-stage process using the package glmtmb in R and with beta binomial error structure. The random effects for each model were line ID nested within operational area, which is sort of the maximum bounding polygon for all operations that occurred at a site, and had a crossed effect operational ID, as operations could occur at different times of the year, had different kill rates, and were spatially different from time to time. And the response in all models was rat traps. So first I ran 10 simple models, which included traps as a function of months since treatment, plus operational size, shape, straight line distance, and least cost distance and cost, and then ran those same set of models with those factors as interactions rather than additive effects. I also included a null model with tracks as a function of months since treatment. So the preferred model to emerge from this process was one that included a time since treatment times least cost, so the accumulated cost of traveling down those shortest paths, and the cost surface on which this best model was based had a 25-fold penalty for lakes, sea and areas over 1,000 meters, and a 1-fold penalty for every elevation 100-meter elevation gain, and a 2-fold penalty for lakes and rivers. Treatment size and shape models were indistinguishable from the null model. I should also say that this best fit model had a delta AIC with the next best fit model, which had months since treatment and least cost as additive terms rather than interactive terms, and was clearly distinguishable. So then to stage 2, I took the best model from stage 1 and then tested this with additive combinations of the parameters elevation, season, and delta T, which are variables we know from the literature have a strong influence on patterns of rat abundance. Delta T, for those who haven't encountered it, which I imagine is a small group, is based on a bit of work by Dave Kelly and is used as a predictor for beach mast. So think of it, I guess, as a proxy for available food. The preferred model to emerge from this model selection process was one that included all parameters. It was clearly distinguishable from the next best model, which was identical but did not include delta T. It was based on a cost surface with a 25-fold penalty for high elevation areas in sea and a 25-fold penalty for lakes and rivers. So to illustrate the model, I created this plot. Down the y-axis, we've got all the parameters included in the model, and the points and whiskers are the model coefficients and confidence intervals. So you can see that months since treatment had a strong positive effect on mean tracking rates. Least cost had a negative effect. Elevation was associated with reduced tracking. Summer and autumn positive. Interestingly, delta T was associated with a negative or a lower rates of tracking, which I find surprising, but I guess can be explained by the fact that we don't treat at random, but most treatment is coincident with MAST, and so you get this negative effect from delta T when normally you might expect a positive one. And the months since
3 minutes,
treatment,
James.
Yeah, got ya. And least cost interaction were positive. I find interactions kind of hard to think about without visualizing them, so I did that in this plot here, which shows that shortly after treatment, at areas that are easiest to get to, those with the least cost, have higher tracking rates than those that are hardest to get to. But this relationship swaps with time, and areas that are furthest or hardest to get to have higher tracking rates, and those that are easiest to get to have lower tracking rates, and this is depressed by elevation. I suspect what's going on here is much as Susan described, that food stocks build up with time, where rats have taken longer to get to and so have higher carrying capacity by the rats finally get there. But it may also be explained by the slower recovery of possums and stoats to these areas. So in summary, as we get better at large-scale rat control, immigration will become an increasingly important driver of rodent recovery. Least cost methods are useful for understanding patterns of rodent recovery, and we could use these models to optimize the geometry of our operational areas to increase treatment persistence. We may also be able to use these models to target rat control to immigration pathways and slow immigration. Thank you.